Hi, everyone. I'm Dr. Rebecca Panko, and I'm a postdoc research associate at the Joseph's Lab in the Department of Plant Biology at MSU. And I'm here today to discuss a bit of my dissertation work, which is evidence of local adaptation of Capsella bursa-pastoris in New York City. Urbanization is increasing worldwide. This is a heat map showing urban population size in 1950. And if we were to compare that with the same map in 2020, we see that not only have areas that were urban expanded, but there's also new areas around the world that are considered urban. And when you have urban areas, this has a dramatic effect on the landscape because cities have what's known as high proportion impervious surfaces. So impervious surfaces are those that prevent water from entering the soil, as you can see here on the left. And if we were to take a land cover map of impervious surface and put that on top of this map of New York City, with impervious surface shown in red and dark red, we can see there's quite a dramatic increase of impervious surface towards the urban core. And when you have this increase in impervious surface, it brings about a syndrome of environmental changes. For example, habitat fragmentation and degradation due to the increase in built infrastructure that correlates with increased human population density, but also altered abiotic and biotic factors. For example, in cities, there is increased water stress and also higher relative air temperatures and other things. Despite all of this, there are some plant species that have reached global distributions and have been successful in urban areas. And we now have some evidence that some plant species are adapting to life in cities. We have some evidence of this, but there's still some key questions. For example, what environmental variables or combinations thereof are serving as selective pressures in cities? So traditionally we use things like human population density and proportion impervious surface, but these are really proxies of urbanization. If we really want to start to pull apart local or adaptation in urban environments, we need to consider things that have a direct effect on plant success for so these environmental variables. This will allow us to answer questions such as, are populations adapting to urban locales at large, such that we have urban and rural populations? Or are they locally adapting at fine spatial scales within cities due to the heterogeneous nature of urban environments? So as part of my dissertation work, I quantified the effects of natural selection in shaping Capsella bursa-pastoris populations in New York City. Look to see if there's evidence of local adaptation, and if so, what environmental variables are potentially driving that. And I tested this using a reciprocal transplant experiment and a foreign local analytical approach. Capsella bursa-pastoris, or shepherd's purse, is a very common weed worldwide. It is a close relative of Arabidopsis thaliana, the model plant organism. Shepherd's purse is an allotetraploid. It predominantly self-pollinates, it's very short in stature, and it can produce copious amounts of seed. In urban environments, you'll find them in these kind of disturbed sites, so street tree pits, sidewalk cracks, roadsides, things like that. I collected seeds from 360 plants representing 24 New York City populations, shown here in black dots. And from those 24 populations, I selected 8 sites to use for a reciprocal transplant experiment. And they based that on their local impervious surface, the safety and accessibility to the sites, and the distance apart to minimize relatedness of the pairs. So here on the right is a map showing my reciprocal transplant sites with the colors indicating paired sites. Here's an example of a pair. This is Site 3 and Site 4, which are shown in yellow on the map. Site 3 is in Yonkers, and Site 4 is in Williamsburg, Brooklyn. I grew seedlings in the lab and then transplanted them to the reciprocal transplant sites, about 20 seedlings per site, 10 from the origin site and 10 from its paired site. And on the day that I planted them, I recorded a bunch of environmental conditions, including soil compaction and moisture, heavy metal content of the soil, bare soil cover, organic litter cover, so like leaves and things like that, and then tree canopy cover. And then I conducted principal and component analysis to see the relationships between the site's proportion impervious surface and the environmental conditions of the site. And once I did that, I found that PCs 1 through 5 explained 92% cumulative proportion of the variance, and that PC1 explained 28% of the variance, and that was positively correlated with impervious surface. And PC2 explained 22% of the variance, and that was positively correlated with soil copper, vanadium, and sodium. So if we just look at those two and compare those, what I want you to notice is that things that we consider kind of urban syndromes clump together, right? So lead in soils, bare soil cover, soil compaction, and then of course impervious surface. Whereas things that are maybe not thought of as urban syndromes, such as canopy cover and litter cover, meaning organic litter, fall away from those. Using the principal component analyses, I did a mixed model ANCOVA to determine the effects of local plant and foreign plant, the environmental variables, so the principal components, and their interactions on plant survival time, flowering, and seed pod production. So I would revisit the reciprocal transplant sites many times and record a lot of different data, including flower number, seed pod number, and mortality. Today, I'll show my results for seed pod number. So this is the results for a single model showing on all of the y-axes you have seed pods produced, and on each x-axis you have the principal components that I mentioned. So if we look here, we can see that local plants produce more seed pods than foreign plants. Local plants here are shown in red, foreign plants here are shown in blue. And particularly at the average values of these principal components, or when x is 0, the red line is always above the blue line. What selective pressures are driving this result? If we just look at principal components 2 and 3, we see here that both plant types, right, foreign plants and local plants, are responding similarly to the selective pressures, but that local plants always have more seed pods across these adaptive landscapes. If we were to look just at PC1 and PC4, here local plants and foreign plants have a different response as the selective pressures increase. And for all of these, you can think of the selective pressure increasing towards the right. And local plants are producing more seed pods as impervious surface increases, as shown in A, and as cadmium, arsenic, and selenium increase, as shown in D. And lastly, if we look at PC5, here both plants are responding similarly to high levels of lead and chromium, with local plants producing more seed pods at low amounts of lead and chromium in the soil. However, I should mention that of my 8 reciprocal transplant sites, 7 of them had lead concentrations that exceed the toxic range known to plants. So when I say low, it's still pretty high amounts of lead. So are Capsella bursa-pastoris populations adapting to urban locales at large? Well, yeah, because there is evidence for local adaptation. Local plants produce more seed pods than foreign plants, both as impervious surface increases, but also at all the average values across the PCs. Are they adapting at finer spatial scales within cities? Well, yeah, because there's a bunch of site-specific heavy metal content, and the local plants are producing more seed pods than foreign plants in sites polluted with those heavy metals to a point, right? Because when you get to the lead results, once you hit a certain threshold of lead, everyone kind of performs the same. Currently, I'm working at the Joseph's Lab to establish a genetic basis for the observed differences in fitness, and stay tuned for a manuscript with this work, which I'm currently working on. With that, I'd like to thank Joseph's Lab and the Department of Plant Biology at MSU, my grad team at NJIT, Dan Bunker Lab, and other collaborators. If I don't get to answer your question today, you can feel free to email me at this email address here, or reach me here on social media. So with that, I'd like to thank you.